Well, thank you, Mark. Thank you, Carl, and all the organizers for inviting me. It's a real pleasure to be here this morning and to be part of this exciting meeting about genetics and diseases. And what I'd like to talk to you about is the progress and the development from the crazy kids at the Human Cell Atlas, and also a few thoughts basically about the future and about connections between our community and this community. So just in case you're not aware, very quickly, the basic idea of the Human Cell Atlas is to make a comprehensive, unbiased, and molecular resolution map of the human body. So to basically use the new methods that we've heard a little bit about this morning that have been evolving over the past 10 years, kind of a pace, I would say, that are single-cell RNA sequencing and spatial methods, both sequencing-based and imaging-based, for mapping tissues at a kind of Google, what we sort of think of as a Google Street Maps resolution. In other words, the molecular fingerprints of our cells, as opposed to a more coarse-grained mapping of our tissues. And so why are we so interested in cells? Well, because they are really the fundamental units of our bodies. They make up the tissues, which contribute to our organs and our organ systems. And so if we want to get to that very high resolution view, we need to understand these basic units. And as I said, the real, I would say, catalytic technologies that kind of made this idea sort of bubble up in various places are single-cell genomics that has really exploded in terms of technology development. I'll mention that in the next slide in a minute. And then the spatial methods for mapping cells and indeed single molecules in situ, in tissue sections and tissue consecutive sections in 3 dimensions. So this is a kind of overview. I'm sure you've seen this type of thing before, where we have the timeline and mapping the explosion of the highly parallel large numbers of cells here on the y-axis on a log scale that are captured by experiments. And this has really been, let's say, roughly, if we go back to Azim Sourani's papers as the first papers for sequencing of transcriptomes from single cells. If we kind of use that as our rough marker, it's really a 10-year development that's gone over several orders of magnitude, where we're now capturing sort of hundreds of thousands and millions of cells in single experiments. And this is partly due to molecular biology, and it's partly due to the cell capture technologies. And together, these things have really pushed the methods, to a point where I would say they are human genetics ready. At the same time, maybe lagging a little bit behind in terms of the pace of development are the spatial technologies, but these are super exciting because they really allow us to map cells and individual genes, proteins, or RNAs in situ, and they can be either sequencing-based, like these spatial transcriptomics and FISSEQ technologies, or they can be imaging-based or actually mass spec-based, interestingly. And what this shows is that there are different sort of trade-offs between tissue area, the field of view that you're capturing with these in situ methods, and the plexity of the method in terms of the number of molecules. And this is a slide put together by Aviv Regev, my partner in crime in this adventure over the past few years, where you can sort of see how the spatial methods span an RNA, nucleic acid, and protein, and they're basically different angles for tackling this problem, different technologies that are evolving. And basically in human cells, we unite these different technologies, different projects, and different investigators use different combinations. And of course, what we need to apply them to are human tissue samples. And so this has been a kind of interesting journey, kind of for me as a basic scientist, which is how do we get these tissue samples? How do we work with the clinicians and in our community to acquire samples for this molecular mapping endeavor? And this kind of summarizes the different types of samples. I'm sure you've had discussions here about the biobanks and the different tissue acquisition sources. And of course, they encompass either biopsies, which living donors, really, and probably the freshest end of the spectrum, and can include sort of control reference samples as well as disease. Deceased transplant donor tissue, which allows very deep and very precise sampling that you can't get from biopsies. It also includes human development for us, so that's tissues from the Human Developmental Biology resource that are from social terminations from about 3 to 20 post-conception weeks. And that's very exciting because we have access to that black box to a large extent, where development has mostly been studied in model organisms, and we can now access it really in human tissues. And then sort of at the least fresh but most comprehensive end are post-mortem examinations, so warm autopsy type scenarios. And so these are tissue acquisition sources that we've been using in Human Cell Atlas. Here in the UK, we have the Human Developmental Biology resource, funded specifically for the Human Cell Atlas by the MRC recently, and also the Cambridge Biorepository for Translational Medicine, funded for the Human Cell Atlas specifically by both Wellcome and the Chan-Zuckerberg Initiative. And so that's really, really key kind of at the very beginning of the pipeline to acquire the tissue, then apply the technologies, and of course, then move on to the computation. And so what sort of progress have we made kind of over the past few years? So Human Cell Atlas only started a couple of years ago, and you can see that there's been good kind of and tighter, increasingly good communication around biological networks, like, for instance, the kidney, where you can see here data from a UK consortium that I'm involved in, comparing healthy kidney to tumor samples, developmental atlas approaches from us and other labs in the US, and then kidney tissue and organoids from Boston here, from Aviv and Agreka and Orit. And this kind of, I think, biological network, or this kind of coordination among people who are interested in similar biological questions may also be relevant for this consortium. And that there's the coordination element is something I'll come on to in a second. The developmental cell atlas here, I've mentioned before that we can actually access human developmental stages, which is incredibly exciting. And here's an example of our work on the deciduous placenta, the maternal-fetal interface, which is really fascinating from an immunology point of view in terms of tolerance of paternal alloantigens. Fetal liver hematopoiesis, again from the UK, and then here's a heart example with in situ sequencing from Sweden. So these are basically just to show you that the Human Cell Atlas is happening. Data is being generated. There will be these very high-resolution maps at increasingly dense kind of coverage of the body, and this can provide a reference, for instance, for then going up to the human genetic level. So in terms of the kind of pipeline, the data coordination, the data crunch, the crunching and munching through the data is really at the end of this pipeline of tissue acquisition, data generation by these high-resolution technologies, and then the data analysis. But what's really interesting in our consortium, I think, kind of from a science history or whatever perspective, is that this is actually an activity that was right at the beginning of the formation of the consortium, and that's really thanks to the Chan-Zuckerberg Initiative, Corey, who's here, who saw the potential and saw the importance of this, and invited a proposal for building a collaborative data coordination platform that's distributed across several centers. The EBI here, Yoon didn't mention it, but there's a team on our genome campus in Hingston involving some part of Sanger and a lot of EBI, and also at the Broad and at UCSC and the Chan-Zuckerberg Initiative itself. And this has actually been a really great kind of nucleation point for the consortium, because it also forces people to think about standards, coordination across several consortia, harmonized data processing pipelines, metadata, and so on and so forth. And you can see the team here that involves more than 50 people. So sort of one step back, what's the governance structure of the consortium? And in fact, this organizing committee that's global and involves over 30 members now, and that's chaired by Aviv and me, that was also formed pretty early on in the initiative and has been exceptionally helpful in developing the thinking around coordination of the consortium. And the other thing that I would say is what's been absolutely crucial is having executive offices with staff members who can support the logistics of organizing meetings, taking notes, making agendas, and so on and so forth, and making the website, having a project registry, and so on. We've got working groups that we set up kind of along the way. The governance of the data coordination platform is obviously crucial, as is the steering committee for the data coordination platform in the different places. A close relationship with the funders and the funders forum has been extremely helpful and productive. And the online tools, basically, so the project registry is at humancellatlas.org/joinhca. You can have a look yourself. We've also, the staff and the executive officers have also supported kind of matchmaking activities for consortia, for people with similar interests to join up or sort of complementary interests. There are lots of tools nowadays like Slack and protocols.io that make communication across the globe feasible. And this is, I should say, like I hope this consortium will be, it's a completely international project that involves thousands of scientists from over 40 countries. You can see the newsletter. There are also old-fashioned communication media, like a newsletter that's gone out to 1,000 members. And all of this is really, this incredible progress has really been possible through this very close partnership with the funders. And what it has meant is that I estimate that across the philanthropic funders, the Chan Zuckerberg Initiative, the Wellcome, Helmsley, Manta, and so on, and the government funders, the NIH, the EU, the UK Medical Research Council, the Swedish government funders, and French government funders, and so on, that altogether we're now probably in the order of $200 million that have been committed to the Human Cell Atlas. And that's over a period of really 2 years. And so that has been a mind-blowing success and tremendous rate for this project. It also poses its own challenges, and some of those we didn't anticipate, but we're kind of dealing with them along the way. And one of those is that how do you integrate across these consortia, some of which are completely international because the funder doesn't have any boundaries in their sort of charter, like the Chan Zuckerberg Initiative, but some of them are national because the government funders are kind of more nationally based. And so how do we basically bring all of this together and kind of shepherd these different consortia towards a common goal of creating this comprehensive resource? And this is something we're actually currently grappling with. I don't have the full answer, but some of the strategies that we're planning to use for this are the project registry, to link related projects through the biological networks, which are, you saw the one for the kidney, for human development, and so on, to basically bring people together, to organize meetings in person and through teleconferences that span across the consortia, because they all have their own meetings, obviously, but we need to kind of link them together. Possibly to have coordination in terms of annotation, like data annotation, including actually a common coordinate framework for the human body and for the tissues. As I said, the data coordination platform is a really good nucleus because this provides basically something I think many funders and many consortia can agree should be centralized. And then also tissue acquisition and technology hubs could be ways of bringing the consortia together. And so that's really the end of what I wanted to say about sort of the form and history and lessons from Human Cell Atlas. I hope that we have some advances. I just want to have one more slide kind of looking forwards, and that is how could human cell atlas interact with human genetics. And I think that, as Lude mentioned, and I completely agree with, that the cell atlas technologies are ready for human genetics. And so really the interaction between human cell atlas and consortia like these could be very natural and productive. And so in my mind, basically, in 5 or 10 years' time, if we look forward, there may be millions of human cell atlases. So we really have not just one reference human cell atlas, but a sort of precision, personalized view of human cell atlas. And that's what I'm trying to say here. And that then allows genetic analyses. And likewise, there are resources, like the Human-Induced Pluripotent Stem Cell Initiative, which is really one of the reasons why I moved to Sanger, because I thought this was incredibly exciting. That there are hundreds of skin and blood samples that have been connected from mostly Cambridgeshire donors, and they, the iPS cells, then can obviously generate sort of in vitro models for many different natural cell types that can be interrogated genetically. And so this, I see real potential here from the single cell side of things, which can help us understand regulation of gene expression at a very high resolution level. It can help us to understand biological processes. So I don't really see this as so much as networks, as more basically modules of cell state that can be determined genetically. And on at our recent retreat, basically, Anna Cuomo, who's a brilliant PhD student from Ollie's group, kind of presented a beautiful framework for a linear mix model to interrogate single-cell genetic associations with various features and processes that can be mined from the data. And this is a version that we're using for also many ex vivo cell type interrogation. So I hope that there'll be fun discussions here about ways that we can interact, not just in terms of governance and organization, so on and so forth, but also scientifically. Thanks for your attention. I'll take any questions.
Thanks, Sarah. That's fantastic. Very helpful for all of us to see, having been as familiar with it. So, questions? Siska?
Beautiful story.
Your name
So Sarah, what can we learn from your success in also getting all the funders involved? Do you have kind of a clear vision, or what was the success story here?
So, I think that this is a great start here to bring everyone together and open the discussion. I would say there are different categories of funders, and with different levels of, as I mentioned, sort of geographic flexibility and funding mode flexibility and so on. And the philanthropic organizations like the Chan Zuckerberg Initiative and Wellcome and Helmsley and Kavli and so on were organizations that we benefited from immensely in terms of their flexibility and their engagement. And then the government funders are more sort of bound to rules, I would say, and nationally restricted and process bound to processes and so on.
Do you think it has something to do with the excitement about something which has novel technology and kind of goes into territories people have never explored? What could be?
Well, the technologies, I think I put them right at the beginning because they are also what catalyzed the excitement from the scientists, and I think that definitely transmitted to the funders. Probably is that we have these new molecular microscope with genomic technologies, and interrogating the human body, opened up many new discoveries, I think, right from the beginning. So that for sure.
Cecilia, then Judy.
I was going to ask the same question, but from a different angle. If
Okay.
we were to propel you back to the first meeting of HCA of about this size, what would you have done differently? What is the lessons? You should take it over a glass of wine sometime.
I mean, there are a couple of things we would have done differently, but you know, overall, I think achieving a governance quickly with the organizing committee was totally right, the right thing to do. I think the other thing I would say is crystallizing a vision with a white paper in retrospect was really the right thing to do. The one thing I would say that we didn't anticipate and we're still kind of working on is how much work the coordination is. It doesn't just happen naturally. It's a lot of hard work, and that's really only been possible to a large extent, I would say, thanks to the staff members at Broad and Sanger who are employed as support staff, and getting more international. I think one thing I would do differently is trying to get more support for mundane coordination, just very simple, sort of infrastructure support, and having that distributed internationally so that there's really a comprehensive coordination. Maybe that's one thing that we could have done better, and we're still kind of catching up on that. Yeah.
So science and organization.
Yeah.
So
yeah.
In terms of discussions, we've been talking about scale, and we've been talking about reuse of data and primary use of data. So I'm wondering, like, one area in terms of geocoding tissues, one wealth of information is, it's not as easy as radiology, but is kind of clinically used paraffin tissues. It's a massive library.
Yeah.
And potentially easier, but has its challenges.
That's right.
Discussions on that?
Yes, absolutely. We've had a lot of discussions on tissue sources, and so I thought you were going to also say kind of coordinate frameworks and ontologies and so on. But the tissues, the paraffin embedded tissues is just one example, and they're amenable to certain technologies, but not so great for sequencing, for instance. So, but they're certainly great for some imaging technologies.
But have you thought about that in terms of scaling that for relatively large numbers? Because like there are tens of thousands of liver biopsies in health systems.
Yeah.
Tens of thousands of kidney biopsies.
Yes, absolutely. So the biopsies are great for like tissues that are accessible, the gut, the skin, and so on, and that scale is great for genetics. They're not so great for geospatial precision and depth, right? But yeah, so different sources have different pros and cons. Nicole, yeah.
Last question, maybe.
So I think the issue was brought up before, and it's something we're thinking about is the depth of coverage of the transcriptome, right, in terms of transcripts, and in terms of the uses that we can think of, using in a QTL framework. You can think about adding some transcriptional predictors of risk scores, for instance, but we would also like to probe the biology, how genes affect multiple transcripts. And so what is I think where it's going to be the sweet spot at which we'll be able to do studies at scale, but also good coverage of what's the landscape in technology development that will allow us to grow.
So I'm super optimistic. And that's not because I'm an optimistic type of person. I mean, I really, the technologies are developing not only in terms of breadth, like I showed, but I didn't show sensitivity, but they're also really improving in terms of sensitivity. So there was a question from someone before about sort of, accuracy and sensitivity and so on. And I think that will not be an issue. Like, we will get down to sensitivity of detection of single molecules in cells. And I'm also pretty optimistic about cost developments. So there are ways of, we've discussed some of the ways of achieving better affordability using different strategies. And so yeah. you know, the better sensitivity is a combination of, again, the molecular biology, you know, the protocols, the capture technologies, and so on.